Hi, this is one of my presentations for Sunday, July 16th, 2023. It is titled GRIN2B genotypes and APOE haplotypes affect the age and onset of Alzheimer's dementia, as well as variations in dementia staging according to the use of Memantine. Memantine antagonizes the NMDA receptor of glutamate, leading to possible behavior improvement and a synergistic effect with cholinesterase inhibitors on slower function and cognitive decline in Alzheimer's dementia. On the other hand, pharmacogenetic studies are useful to identify genetic factors that participate in the heterogeneity of drug response and may be helpful to bring new insight into the pathophysiology of Alzheimer's disease. The native NMDA receptor consists of a tetramer with 2 NR1 subunits and N2 of 4 known NR2 subunits. The NR2B subunit is a common subunit of functional NMDA receptors, whereas the GRIN2B gene is mapped to 12p12. And the RS3764028 has been associated with variable GRIN2B transcription and human susceptibility to Alzheimer's dementia. APOE is a moderately penetrant gene that is neither a prerequisite nor a sufficient agent for development of later-onset Alzheimer's dementia. So our learning objective was to evaluate the role of GRIN2B genotypes and APOE haplotypes on the age of dementia onset and cognitive and functional response to memantine in patients with Alzheimer's dementia. So this is a prospective pharmacogenetic study in which participants with late-onset Alzheimer's dementia were screened with clinical dementia rating sum boxes, the index of independence and activities of daily living, Lawton scale for instrumental activities of daily living, the mini-mental state examination, the severe mini-mental state examination, and a 15-item clock drawings test. And they were followed for 1 year. We genotyped the RS3764028 SNP of the GRIN2B gene and APOE haplotypes. We correlated these genetic variants with use of memantine or not for test score variations along 1 year. So in table 1, we see the demographic and clinical results. All tests were considerably different after 1 year, with the exception of clock drawing test. In table 2, we can see that the GRIN2B genotypes were in Hardy-Weinberg equilibrium. We had 190 consecutive outpatients, and 141 of them used memantine. APOE E4 carriers had earlier dementia onset, whereas the A allele of RS3764028 had a cumulative effect on later onset of Alzheimer's dementia. And in table 3, we see that memantine protected APOE4 non-carriers who also carried the CC genotype of RS3764028 regarding clinical dementia rating sum boxes variations, but not regarding other test score variations. So we conclude that genotypes of RS3764028 and APOE haplotypes are associated with the age and onset of Alzheimer's dementia, as well as variations in dementia staging according to the use of memantine. Thank you.